Welcome, everyone, to another deep dive. We've got a really fascinating stack of material today.
Yes, we do.
We're focusing on a molecule with a, well, truly unique history, and it has some potentially remarkable implications for age-related and neurodegenerative brain conditions.
Mm-hmm.
If you're someone who values detailed, rigorous scientific exploration, the kind that gets into the why and the how, not just the what,
Mm-hmm.
then this deep dive is definitely for you.
Indeed. The source material we're really unpacking is a comprehensive review. It's titled Palmitoylethanolamide, PEA, in age-related and neurodegenerative brain conditions. A comprehensive review of historical development, mechanistic insights, and therapeutic promise.
Right.
So it offers a deep dive, you could say, into the scientific journey of palmitoylethanolamide, or PEA, all the way from its, well, frankly unexpected origins to its potential role in tackling diseases that currently have pretty limited therapeutic options.
Okay, let's unpack this then. Our mission for this deep dive is really to trace that scientific trajectory. We want to understand not just the PEA has shown promise, but how it actually works, why that mechanism is significant,
Which is crucial.
and what the current evidence across the whole spectrum of research, from foundational discovery to modern clinical trials, actually says about its effects. We're talking devastating conditions like Alzheimer's, Parkinson's, ALS, frontotemporal dementia, stroke, and mild cognitive impairment.
Exactly.
We're aiming for that granular detail, the kind you demand to feel truly well-informed. So where does this story even begin? Because it's not in a typical drug discovery lab, is it?
No, not at all. It's a really surprising origin story, actually. It starts with clinical observation way back in the early 1940s. Physicians at the time, they were working with underprivileged children and noticed something quite, well, unexpected.
What was that?
Well, they observed that supplementing the children's diets with dried chicken egg yolk seemed to somehow reduce the recurrence of rheumatic fever.
Really? Just from egg yolk?
Yeah. And the interesting part was, this was happening even when the children were still exposed to the ongoing streptococcal infections that actually caused the fever in the first place. It was a curious empirical observation. Didn't immediately make sense, maybe, but it was significant enough that it warranted further investigation.
So seeing this effect in the clinic, that sparked a scientific search for what was actually in the egg yolk that was responsible, made them think, okay, what is this stuff?
Precisely. That led researchers throughout the 1950s to try and isolate the active component, and they found that purified lipid fractions, not just from egg yolk, interestingly, but also from other common foods like peanut oil and soy lecithin, they exhibited anti-allergic and anti-inflammatory properties in animal models.
Okay, so they were narrowing it down.
They were, and this systematic work culminated in 1957.
And that's when they finally identified it, nailed it down.
Yes. A team led by F.A. Kuhl Jr. at Merck & Co. successfully isolated and identified the specific compound. They named it N2-hydroxyethylpalmitomide, which, as you said, is a bit of a mouthful.
It just rolls off the tongue.
Isn't it? We now universally know it as palmitoylethanolamide, or just PEA.
Much easier.
Much. And those initial isolations confirmed its presence in those key dietary sources. Soy lecithin, egg yolk, peanut flour, just as their earlier experiments had sort of hinted at.
Okay, so from dietary observation to isolation and identification. At that point, it seemed like an interesting compound found in food. What happened next? How did it transition from just being a dietary component to something the body might naturally use?
That's where a really crucial finding came in back in 1965. Researchers led by Butcher, they discovered that PEA wasn't just something you consumed in your diet. It was also present in mammalian tissues.
Oh, okay, so our bodies make it.
Exactly. This confirmed it was an endogenous molecule, meaning our own bodies actually produce it. And that discovery immediately suggested PEA had some kind of natural physiological role. It moved it beyond just being a dietary curiosity.
And that endogenous discovery, that must have led to it being looked at clinically, right? I think I read about something called impulsin.
Yes, exactly. That finding spurred clinical investigation, particularly in what was then Czechoslovakia. By the 1970s, PEA was actually marketed as impulsin by the pharmaceutical firm Spofa.
It was used quite a bit.
It really was.
Yes.
It administered quite widely to thousands of people, including children, soldiers too, primarily to help manage influenza and acute respiratory infections. Clinical trials conducted back then showed evidence of its efficacy. reducing symptoms and duration, and also a favorable safety profile. There was even a similar product, Palmitoylethanolamide, introduced in Spain around the same time.
So it had this period of seemingly successful clinical use, apparently safe and effective, and then according to the source material, research just stopped. There's this period of scientific stasis mentions for the 1980s. That seems really counterintuitive if it was actually working.
It really is one of the most fascinating parts of its history. You're right. Despite documented clinical success, despite apparent safety data from treating thousands of people, research into PEA genuinely waned for over 2 decades.
Wow.
And it eventually fell out of widespread clinical use. Now, the review is very clear on this. It wasn't because of findings of toxicity or lack of efficacy in those early uses.
So what was it then?
The critical knowledge gap was fundamental. They failed to identify a specific molecular target or, maybe more importantly, a plausible mechanism of action.
Ah, okay. So they knew that it did something, but they had no idea how it was doing it.
Exactly. Without understanding how it worked at a cellular or molecular level, it just couldn't be easily integrated into the dominant paradigm of drug development at the time, which, was heavily focused on identifying specific receptors and designing molecules to interact with them.
Right, the lock and key model.
Precisely. So PEA remained this compound that worked empirically, sure, but it lacked a clear biological rationale that fit the prevailing scientific framework. This 2-decade stasis, it's a really powerful lesson in the importance of mechanism. Sometimes even compelling clinical results just aren't enough if you can't explain the underlying biology.
So how did it come back from that, from scientific obscurity? Who brought it back into the light?
Well, the review calls it the resurrection, or sometimes the Montalcini Renaissance. And it's thanks largely to the work of the Nobel laureate Professor Rita Levi Montalcini.
Ah, the Nobel laureate. Okay.
Her research group published a truly landmark paper in 1993, and this paper basically provided the missing piece of the puzzle, the mechanism.
Finally. So this is where it gets really interesting.
It truly revitalized the field. They demonstrated that PEA wasn't just acting randomly, it actually functioned as a natural modulator of hyperactive mast cells.
Mast cells. Those are immune cells, right? Key inflammatory players.
Exactly. Key players in the inflammatory response in many tissues, including the brain. Based on this, Montalcini proposed this really elegant concept of an autocoid local injury antagonist, or ALIA for short.
Autocoid local injury antagonist, ALIA. Got it.
Yes. And the idea is pretty straightforward. PA is an endogenous substance, something our body makes naturally that's synthesized on demand right where it's needed in response to injury or stress or inflammation.
Okay.
Its role is essentially to restore a local tissue homeostasis, basically, to calm things down and protect the tissue where the insult is happening. She even coined the term alamide for this class of molecules. It really positioned PEA as part of the body's own intrinsic self-regulating protective system that gets activated during times of local stress.
So it's actually part of the body's natural response to injury and inflammation.
Yeah.
Like an internal first responder.
Precisely. That's a great way to put it. And this elegant mechanism, it finally provided the scientific rationale that had been missing for, decades. It explicitly linked PEA's action to the modulation of these non-neuronal immune cells, like mast cells. And that effectively bridged the crucial neuroimmune axis, that complex interplay between the nervous system and the immune system.
And this was happening around the same time people were learning more about the endocannabinoid system.
Yes, exactly. The discovery occurred alongside the increasing understanding and discovery of the endocannabinoid system, to which PEA is functionally related. All of this together just reignited global scientific interest and really paved the way for the modern research we're going to discuss today.
So the history really underscores that principle. Knowing how something works is just as critical, maybe even more so for long-term scientific and clinical adoption, than just knowing that it seems to work. The Impulsion era showed clinical effectiveness, apparently, but the lack of mechanistic insight led to its decline. And then Montalcini's discovery provided the biological rationale needed for its modern resurgence.
That's exactly the takeaway emphasized by the review. The early clinical use was purely phenomenological, describing the effect without explaining the cause. That 2-decade stasis really highlights the scientific community's need for a biological rationale to drive further research and integration into clinical practice. It was only the ALIA concept that made PEA a legitimate research candidate again, and that explains why research is accelerating now, decades after its initial identification.
And the review includes a really helpful timeline of these key events, doesn't it, showing that progression?
Yes, it lays it out clearly. You can see that arc. The 1940s with the observation linking egg yolk to rheumatic fever protection. Then the 1950s bringing the isolation and identification of PEA itself. The 1960s showing its endogenous, made in the body.
Then impulsin in the 70s.
Right, its clinical use as impulsin. Then that puzzling 1980s stasis due to the lack of mechanism, followed by the pivotal 1990s with Montalcini's ALIA concept, linking it to mast cells and neuroimmunity.
And then the modern era.
Exactly. The 2000s and onward, where renewed interest, coupled with new discoveries about its targets and, crucially, advances in formulation, drives the clinical trials we're about to get into. It's really a fascinating scientific journey.
Okay, so with that incredible history and the crucial understanding provided by Montalcini's ALIA concept, let's dig into how this molecule actually works at a deeper level, particularly in the context of neuroprotection and neurodegenerative diseases. You mentioned it's not a simple one-target drug.
That's absolutely key. The source material describes PEA as a pleiotropic lipid mediator. Pleiotropic just means it has multiple effects, acting on many molecular targets and pathways at the same time. It's not a single key fitting a single lock. Its fundamental role as an ileamide is that of an endogenous homeostatic regulator. You can almost think of it as part of the body's own finely tuned system, mobilized when there's stress or injury, specifically to restore balance, resolve inflammation, and protect cells from damage.
So the body actually makes more of it when it's under duress, when things go wrong.
Precisely. That foundational LEIA concept states that PEA synthesis is naturally upregulated, increased in tissues that are experiencing stress or injury. This is the body's own built-in protective mechanism kicking into gear.
And there's evidence for that.
Yes, absolutely. This is supported by evidence showing that PEA levels have actually been found to increase in the brain in conditions like Alzheimer's disease, Parkinson's disease, and also following traumatic brain injury. This pattern supports its role as a natural counterresponse mounted by the body against the ongoing pathology. It's really a different paradigm than most pharmaceuticals, which often, impose a specific blockade or activation on a pathway. PEA seems to work more by enhancing the body's own pathways for resolving issues and restoring balance.
That distinction feels important. It's working with the body's systems, not just overriding them. So what are these multiple molecular targets and pathways that contribute to its neuroprotective effects?
Right.
How does it do what it does?
Right. The review details several, involving both direct and indirect actions. Probably the most primary and arguably the most well-established mechanism is its direct binding to and activation of a nuclear receptor called peroxisome proliferator activated receptor alpha, or PPA alpha for short.
PPAR alpha.
Yeah.
Okay, we've heard about nuclear receptors before. They often get involved in regulating genes, right?
Exactly. PPAR alpha is a transcription factor. So when it's activated, it binds to DNA and regulates the expression of specific genes. It plays a critical role in processes like lipid metabolism and, crucially for neurodegeneration, inflammation.
How does activating it help?
Well, when PEA directly binds and activates PPAR alpha, it triggers a cascade of events that leads to the suppressed production of pro-inflammatory proteins, things like certain cytokines, those signaling molecules that really drive inflammation. And at the same time, it enhances pathways involved in cellular protection and survival.
Yeah, the source provides pretty compelling evidence that this PPAR alpha activation is actually essential for PPA's benefits in brain conditions, doesn't it?
Absolutely. The review highlights data from animal models of Alzheimer's disease that make this connection, well, unequivocally clear. In AD mouse models that were genetically engineered to lack the PPAR alpha receptor, the neuroprotective effects that are normally seen when you give them PEA were completely absent.
Completely gone.
Completely gone. If you remove the target, PPAR alpha in this case, and the therapeutic effect vanishes, it provides strong, really definitive evidence that this target is essential for the observed action. It's a direct mechanistic link.
That's a crucial piece of data, especially for clinicians trying to understand the why.
It really grounds the biological rationale. Now, beyond this direct action on PPAR alpha, PEA also influences the endocannabinoid system. But it does this in a more indirect way, often referred to as the entourage effect.
Okay, I've heard that term before, often related to cannabis.
Yes, and PEA is structurally related to endogenous lipids like the endocannabinoid anandamide. It's sometimes categorized as a cannabimimetic or an N-acetylethanolamine, or NAE.
So it looks a bit like cannabis compounds, chemically speaking, But it doesn't act quite the same way.
Precisely. That's a really important distinction to make. It is not a classical cannabinoid like THC or CBD. PEA has very weak, almost negligible affinity for the primary cannabinoid receptors, CB1 and CB2.
So how does it influence the system then?
Its influence comes indirectly. It works by inhibiting an enzyme called fatty acid amide hydrolase, or FAH.
And FAAH, that's the main enzyme that breaks down anandamide, the body's own endocannabinoid.
Yes, exactly. So by slowing down the activity of FAAH, PEA essentially increases the levels of the body's own anandamide that are present, and it prolongs anandamide's action at its own targets, including the CB1 and CB2 receptors.
Ah, I see.
So it contributes to the endocannabinoid system's overall anti-inflammatory, analgesic, and neuroprotective tone, but it achieves this by boosting the effects of the body's own anandamide rather than PEA directly binding to those main cannabinoid receptors. That's the essence of the entourage effect in this specific context, enhancing the activity of other beneficial endogenous compounds.
Okay, so direct action via PPR alpha, indirect action via the endocannabinoid system by inhibiting FAH.
Yeah.
Are there any other notable targets mentioned?
Yes, the source also mentions that PEA can directly activate a couple of orphan G-protein coupled receptors, specifically GPR55 and GPR119.
Orphan receptors, meaning we don't fully know what they do yet.
Pretty much. Their exact roles in neuroprotection are still being fully mapped out, but they seem to be implicated in signaling pathways related to metabolism and inflammation. There's also mention that PEA might act as a positive allosteric modulator of the TRPV1 channel, which is involved in pain signaling pathways.
So it really does hit multiple targets.
It does, and this multi-target engagement really helps explain how PEA can exert such broad effects across different cell types and hit multiple aspects of complex pathologies like neurodegeneration.
And focusing on those cell types for a moment, the review strongly emphasizes how PEA tackles neuroinflammation, specifically by modulating non-neuronal cells. We've heard increasingly that neuroinflammation is a key driver of these diseases, not just some side effects.
Absolutely. That understanding has really shifted. Chronic low-grade neuroinflammation is now understood to be a unifying pathological feature across virtually all age-related and neurodegenerative conditions. It's no longer considered just a passive consequence of neurons dying. The source material clearly states it's a crucial driver of the degenerative process itself. And this damaging inflammation is primarily orchestrated by the non-neuronal cells in the brain, specifically the glial cells. So PEA's main neuroprotective strategy appears to be quelling this harmful inflammatory environment by modulating the activity of these crucial support cells.
So it targets microglia and astrocytes, the brain's resident immune cells.
Precisely those. Microglia and astrocytes are the main immune defenders in the central nervous system, but in pathological states, they can become overactivated and release a storm of pro-inflammatory mediators, things like TNF-alpha and IL-1 beta, which are key cytokines, and also reactive oxygen species, ROS, which cause oxidative damage.
And that's toxic to the nearby neurons.
Exactly. It's toxic to the surrounding neurons, and it perpetuates this cycle of damage and inflammation. PEA, primarily through its activation of PPAR-alpha, has been consistently shown in preclinical models to effectively down-modulate this harmful glial activation.
So it calms them down.
It helps shift these cells away from that aggressive pro-inflammatory state towards a more quiescent or maybe even a neuroprotective phenotype. And by doing that, it reduces the collateral damage to neurons. This glial modulation is seen as central to the observed benefits of PEA in animal models of Alzheimer's, Parkinson's, and stroke.
And just connecting back to Montalcini's original work, it still acts on mast cells in the brain as well.
Yes, that initial insight holds true. Mast cells also reside in the brain and surrounding tissues, and they can degranulate, releasing inflammatory substances. PEA effectively inhibits their activation and degranulation, disrupting another pathway contributing to neuroinflammation. It really brings the story full circle back to Montalcini's foundational work on those cells.
Okay, so we have this endogenous molecule, PEA, with a really sophisticated multi-target mechanism. It involves PPAR-alpha, the endocannabinoid system via FAH inhibition, and direct modulation of key inflammatory cells like glia and mast cells. It sounds fantastic on paper, but there's often a significant hurdle between discovering a molecule and getting it to work effectively in the body, especially when taken orally. A source talks about the formulation imperative. This sounds critical.
Oh, it's absolutely critical, especially for clinicians considering this molecule in practice. And it's a point the review stresses repeatedly. The biological discovery of PEA's mechanism, as powerful as it is really only half of the story of its modern clinical emergence. The molecule PEA itself, being a long-chain fatty acid amide, is highly lipophilic, meaning it's fat-soluble.
Which, as you hinted at earlier, makes it difficult for the body to absorb effectively from the gut, right? It doesn't like water.
Exactly. It is practically insoluble in water. And that leads to very poor oral absorption and really limited bioavailability when it's in its naive, raw, crystalline form. This was a major practical limitation that almost certainly contributed to its historical disappearance from widespread clinical use.
Right.
So the remarkable clinical results we're seeing and discussing today are, to a significant extent, the direct result of applying advanced pharmaceutical technology to overcome this fundamental solubility and absorption problem.
So just taking raw PEA powder, for instance, might not yield the same results at all compared to taking a specifically formulated version.
Fundamentally, no. The source material emphasizes this very strongly. The positive clinical outcomes we're seeing are a property of the formulated PEA product, not just the PEA molecule in isolation. This is a vital point for interpreting the evidence. When you're evaluating clinical studies, you must pay attention to the specific formulation used, because results from naive PEA are unlikely to be comparable to those from its bioenhanced forms. It's really a synergy of the molecule's biology and the technology that allows it to reach its targets effectively.
That's a really important practical point. So what are the main types of these advanced formulations that make it work better?
Well, the most significant technological advance described has been micronization, and even more effectively, ultramicronization. This involves using processes like jet milling to dramatically reduce the physical particle size of the raw PEA material.
How small are we talking?
We're talking from potentially hundreds of microns down to particles less than 10 microns for micronized PEA and PEA, and even smaller, often submicron, for ultramicronized PEA. PEA.
So it's the same chemical, just ground down incredibly fine.
Correct. This doesn't alter the chemical structure at all, but what it does is vastly increase the total surface area of the material that's exposed to the fluids in the gut. And this increased surface area is key because it significantly improves the rate at which it dissolves and consequently its absorption through the gastrointestinal tract.
Makes sense. More surface area, faster dissolving.
Exactly. Studies clearly show that ultramicronized PEA, in particular, demonstrates superior oral efficacy and leads to much better absorbability compared to standard naive PEA. And this allows it to achieve therapeutically relevant concentrations in target tissues, including crossing the blood-brain barrier to reach the brain and spinal cord, which is obviously essential if you're targeting effects in neurodegenerative conditions.
Okay, so ultramicronization, PEA, is key. And then there's the co-ultramicronization with other compounds like luteolin, PEA-LUT.
Yes, that represents a further refinement, leading to formulations often designated as PEA-LUT, or sometimes co-ultrapellet. This approach was designed to address another key aspect of neurodegeneration. While PEA is clearly a potent anti-inflammatory agent, the source notes it doesn't possess significant direct antioxidant effects on its own.
Right.
However, neurodegenerative diseases are typically driven by both chronic inflammation and oxidative stress. Luteolin is a natural flavonoid compound found in many plants that has well-documented strong antioxidant and also anti-inflammatory properties of its own.
So combining them in this special co-ultramicronized way is designed to target both of those key pathological processes simultaneously. Hit both pillars of the problem.
Precisely. Co-ultramicronizing PEA together with luteolin creates what's believed to be a synergistic formulation. The idea is to more effectively target both inflammation and oxidative stress, those two core pathological pillars contributing to neurodegeneration, at the same time. And the review does highlight that many of the most impressive clinical results reported so far, particularly in conditions like frontotemporal dementia and that MCI case study we'll get to, have specifically utilized this synergistic PEA-LUT formulation. It really underscores again how the formulation is integral to the observed biological outcome.
Okay, with that detailed understanding of how it works, its multi-target mechanism focusing on taming neuroinflammation and supporting endogenous resolution, and the absolute necessity of using these bioavailable formulations like PEA or PEA-LU, let's finally dive into the evidence. What does the research from preclinical models right through to human studies actually say about PEA in specific neurodegenerative conditions?
Right. The research landscape here is definitely evolving, and rapidly. It's certainly been spurred by that clearer mechanistic picture and the technological ability to actually deliver the molecule effectively now. However, as you often find in emerging areas, the quality and quantity of evidence aren't uniform across all the different conditions. The source material provides a critical look, which is important. It presents high-quality randomized controlled trials, RCTs in some areas, but then more compelling, yet still preliminary, data in others, and also robust preclinical findings that are still awaiting full human translation. So a nuanced condition-by-condition assessment is really vital.
Understood. Let's start then where the evidence is presented as the most robust in human trials. Frontotemporal dementia, or FTD.
Okay. FTD is a particularly cruel and devastating form of dementia. It often strikes individuals much earlier than Alzheimer's, maybe in their 50s or 60s, causing profound changes in behavior, personality, and language. And crucially, despite the urgent need, there are currently no approved disease-modifying therapies specifically for FTD.
A huge unmet need.
Huge. And it's in this challenging context that PEA, specifically the PEA-LUT formulation, has yielded its strongest clinical evidence to date.
And this is based on a specific high-quality trial, not just observational data.
Yes. The source highlights a landmark phase 2 trial. It was monocentric, meaning done at a single site, but critically, it was conducted with a rigorous, randomized, double-blind, placebo-controlled design, published just recently in 2025.
Okay.
This is exactly the kind of study design clinicians rely on for initial evidence of efficacy, and it investigated the effects of that co-ultramicronized PEA-LUT formulation, the synergistic combination of PEA and luteolin we just discussed.
Right. What were the specifics of that trial? Who was in it? What did they take? And what were they measuring?
Okay, the study included 48 patients who had been diagnosed with probable FTD. These participants were randomly assigned to receive either PEA-LUT, the dose was 700 milligrams of PEA plus 70 milligrams of luteolin, taken twice daily, or a matched placebo for a period of 24 weeks, so about 6 months.
And the primary outcome, what was the main thing they were looking for?
The primary outcome measure was the change over those 24 weeks on a comprehensive scale specifically designed for FTD. It's called the CDR plus NACC FTLD sum of boxes, or SOBI for short.
Okay.
This scale is designed to capture the severity of global deficits, functional decline, and behavioral changes that are characteristic of FTD.
And the results on that main outcome measure, what did they find?
The results were, well, the source describes them as remarkable. Patients in the placebo group experienced the expected progressive decline over the 24 weeks. Their mean CDR FTD SOBI's score worsened significantly by an average of 1.39 points.
Okay, that's the natural progression.
Right. In stark contrast, the patients receiving the PEA-UT formulation showed significantly less worsening. Their mean score changed by only 0.53 points over the same period.
Wow, that's a big difference.
It is. The estimated mean difference between the groups was 0.86 points, and this was statistically highly significant with a p-value of 0.005. The review explicitly states that this finding means the PEA-LUT treatment effectively cut the rate of global disease progression by more than half.
Having the rate of progression in a currently untreatable disease in just 24 weeks, that is genuinely a powerful finding.
It truly is, especially given the devastating and often rapid nature of FTD. And these primary results were supported by key secondary outcomes as well. The PEA-LUT group showed significantly less functional decline as measured by the ADCS-ADL scale. That's a measure of activities of daily living. And they also demonstrated a significant advantage in preserving language function, which was assessed using something called the SAND scale. An earlier, smaller, shorter study had also hinted at benefits in frontal lobe function and behavior, so this builds on that.
And importantly, was it safe? Tolerated well?
Yes, that's crucial. The trial reported it was well tolerated, with no significant difference in the rate or type of adverse events between the PEA-LUT group and the placebo group. So the source concludes that this phase 2 trial represents the highest quality clinical evidence for PEA in any neurodegenerative disease to date, and it provides a really strong rationale for its potential as perhaps the first disease-modifying therapy specifically for FTD.
Okay, so FTD has the solid, high-quality RCT evidence pointing towards disease modification. What about post-stroke recovery? The needs there are obviously huge, too.
Absolutely. Stroke remains a major cause of long-term disability worldwide, and enhancing recovery, both motor function and, critically, cognition, is a significant clinical challenge. Several studies mentioned in the review suggest PEA, particularly that PEA-luteolin combination again, might improve outcomes after stroke.
Is there a large study here, too?
Yes, the largest study mentioned in the review was an open-label clinical trial. It involved 250 stroke patients who were undergoing neurorehabilitation. They received PEA-LUT, same dose, 700 milligram PEA plus 70 milligram luteolin daily for 60 days, so about 2 months.
Open label, though, so everyone knew they were getting the treatment.
Correct. That's an important caveat we need to keep in mind. But within that open-label design, what kind of improvements were reported?
Right, what did they see?
The study reported significant improvements across multiple domains that are really relevant to stroke recovery. Overall neurological status improved, there was a reduction in muscle spasticity, alleviation of pain, and improved independence in activities of daily living.
Okay, broad benefits. What about cognition? That's often severely affected after stroke?
That was one of the particularly striking findings highlighted. There was measurable cognitive improvement. The average score on the mini mental state examination, the MMSE, which is a common global cognitive screen, it increased from an average of 20.2 at baseline to 22.7 within just 30 days of starting the PEA-LUT treatment. The review notes this 2.5-point increase is considered a clinically meaningful gain in global cognitive function, especially in that short time frame.
A 2.5-point increase on the MMSE in just a month post-stroke. That does seem quite rapid.
It is suggestive. And another clinical trial mentioned, this one conducted in patients specifically with acute ischemic stroke, found that adding PCA to standard thrombolytic therapy, the clot-busting treatment resulted in significantly better recovery and cognitive improvement compared to standard therapy alone.
Interesting.
And there was also a smaller study involving just 20 post-stroke patients who had issues with pain and spasticity. It found that adding PEA, in this case 600 milligrams twice daily of just PEA, to their rehabilitation program significantly reduced both of those troublesome symptoms.
So these post-stroke results sound very encouraging, especially those rapid cognitive gains. But as you said, that largest study being open label is a limitation we need to consider.
Yes, it's a significant limitation, scientifically speaking, because both the patients and the clinicians involved knew who was receiving the active treatment. There's a potential for bias. And also, stroke patients naturally experience a degree of spontaneous recovery as their brain heals from the initial injury.
Right, so it's hard to separate the drug effect from natural healing without a control group.
Exactly. Without a double-blind, placebo-controlled comparison group, it's challenging to definitively isolate the specific contribution of the PEA-LUT intervention from that natural recovery process and potentially expectation bias. However, the breadth of the reported benefits, and especially the magnitude and speed of those cognitive gains, certainly provide a strong signal. It underscores the need for rigorous, controlled trials to confirm these promising findings in the post-stroke setting.
Okay, let's move on to Parkinson's disease, PD. What does the evidence suggest there, another major neurodegenerative condition?
Yes, Parkinson's is another progressive neurodegenerative disorder, primarily characterized by its motor symptoms like tremor, rigidity, slowness of movement, but it also involves very significant non-motor symptoms like depression, fatigue, cognitive issues. And while therapies like levodopa are effective at managing the motor symptoms, they crucially do not halt the underlying disease progression.
So the search is on for things that might slow or stop the progression.
Exactly. And the evidence here for PEA, primarily coming from an observational study, suggests that ultramicronized PEA, when used as an add-on therapy to standard care, might have a really profound impact on the disease course itself.
And this is where that really extraordinary finding regarding the Hone and Yar scale comes in, right?
This is arguably the single most extraordinary finding presented in the entire source material we reviewed. Although, again, it's crucial to preface this by saying it comes from an observational study, not a randomized controlled trial.
Understood. But what did they find?
It comes from a study by Bertini and colleagues, published in 2017. They followed a group of Parkinson's patients who were already on stable doses of levodopa therapy. These patients were then given adjuvant ultramicronized PEA. They took 1,200 milligrams per day for the first 3 months and then reduced the dose to 600 milligrams per day, continuing for up to 1 year total.
Okay, and they tracked severity using the Hone and Yar scale.
Yes, the HY scale. It's a standard clinical staging tool for Parkinson's, rating severity from stage 1, mild, unilateral, to stage 5, wheelchair-bound or bedridden.
And the HY scores changed in a, well, remarkable way?
They did. The source highlights the pretrial progression of these specific patients. Before they started PEA, collectively, their average HY score had worsened from 1.8, so relatively mild stage, up to 2.5, showing clear progression.
Okay, that's the expected decline.
Right. Then, after 1 year of continuously taking the add-on PEA therapy, their collective average HY score improved back down to 1.9.
Wait, improved from
Yeah.
2.5 back down to 1.9. That's not just slowing progression. That sounds like a significant reversal of measured severity.
That is precisely the significance that the source draws attention to. Standard Parkinson's therapies like levodopa are fundamentally symptomatic. They manage motor fluctuations, but they are not expected to reverse the underlying disease progression as measured by objective staging scales like the HY scale in this way. This finding suggests not just a slowing, but potentially a near reversal of the functional decline that had measurably occurred previously in these patients. It hints at a deeply neuroprotective or maybe even potentially restorative effect that is, frankly, virtually unprecedented with currently available PD therapeutics.
Wow. That magnitude of improvement on a recognized severity scale is really hard to dismiss, even acknowledging it's from an observational study.
It is. While the limitation of it being observational, lacking a concurrent randomized control group, absolutely must be acknowledged, we can't definitively prove causation. The magnitude of that reported change in the HY score is exceptional.
Were there other benefits too?
Yes, this HY score improvement was also accompanied by broad improvements across a range of symptoms after that 1 year period. The study reported significant improvements in motor symptoms, better performance in activities of daily living, and importantly, significant improvements in crucial non-motor symptoms too, things like mood deficits, fatigue, sleep disturbances, and performance on mental tasks.
So a broad effect.
Yes. But that HY scale finding in particular is such a striking signal. It urgently warrants rigorous confirmation in well-controlled, large-scale, randomized trials to see if this effect is real and replicable.
Absolutely. Okay, what about amyotrophic lateral sclerosis, ALS? Another truly devastating neurodegenerative disease.
Yes, ALS, or Lou Gehrig's disease, is indeed a relentlessly progressive and universally fatal motor neuron disease. It's characterized by progressive muscle weakness, paralysis, and ultimately death from respiratory failure. In ALS, even therapies that can modestly slow the rate of decline are considered incredibly valuable for patients and their families.
Understandably. Is there evidence for PEA here?
There's preliminary evidence, again suggesting the use of ultramicronized PEA, which indicates it may offer some benefit in this very challenging condition. The review mentions initial reports from case studies where PEA treatment led to rapid and notable improvements in things like muscle force, respiratory efficacy, and performance of activities of daily living in some individuals.
Case studies are interesting but limited. Did they mention anything larger, perhaps with more objective endpoints?
Yes, the source does mention a larger study, though the specific design isn't detailed as an RCT, which reported that PEA treatment in ALS patients resulted in a slower decline in respiratory function.
Slower respiratory decline.
That's critical in ALS.
Absolutely critical. And even more pointedly, the review states that in this study, the treatment was found to have delayed the need for tracheotomy and death.
Delayed tracheotomy and death. Those are really hard, unambiguous clinical endpoints.
They are. In a disease like ALS, where the trajectory is so steep and uniformly fatal, impacting these hard endpoints is profoundly meaningful. The ability to extend the time before invasive ventilation, like a tracheotomy, is required, and ultimately to delay mortality, represents a very significant potential improvement in quality of life and a tangible impact on the relentless progression of the disease.
So even if the evidence isn't from a top-tier RCT yet, those outcomes are very compelling. Exactly. While the full details and rigorous design confirmation are needed, these reported outcomes affecting such critical endpoints represent a powerful signal. It aligns with a potential neuroprotective mechanism and strongly justifies further, more rigorous investigation of PEA in ALS.
Okay, let's turn to mild cognitive impairment, or MCI. This is often seen as an early stage of neurodegeneration, sometimes a precursor to Alzheimer's. Is there evidence for PEA here, perhaps intervening early?
Well, according to the review, large-scale clinical trial data specifically for MCI is currently lacking, so we don't have big RCTs here. However, there's one exceptionally compelling single case study highlighted in the source material that is particularly illustrative of PEA's potential in this crucial early stage.
A single case study. Sometimes those can be very powerful if they present really objective, compelling data. What happened in this specific case?
It involved an elderly female patient who had been diagnosed with amnestic MCI. That's the subtype characterized primarily by memory problems, and it's the one most likely to progress to Alzheimer's disease. She was treated with the PELUT formulation, the combination product, for a period of 9 months.
Okay. And the results, clinically.
Clinically, the results reported were quite dramatic. The patient experienced what's described as a significant improvement in cognitive performance, so much so that her formal neuropsychological evaluation, which involves detailed testing of various cognitive functions like memory, attention, executive function, her performance on those tests became almost normal.
Almost normal cognitive function after an MCI diagnosis. That level of recovery is certainly unusual.
It is very unusual. But what makes this particular case study exceptionally compelling, as the source emphasizes, is that this clinical improvement wasn't just subjective. It was corroborated by objective physiological evidence obtained from brain imaging.
Oh, what kind of imaging?
Before starting the PEALUT treatment, she had undergone SPECT brain imaging. SPECT measures regional cerebral blood flow, which is an indicator of metabolic activity. Her initial scan showed the characteristic pattern of hypometabolism, meaning reduced metabolic activity in brain regions typically affected in MCI and early Alzheimer's disease.
Okay, typical MCI pattern.
Right. Then, after 9 months of taking the PEALUT treatment, she had a follow-up SPECT scan, and that scan showed remarkably normalized hypometabolism.
Normalized metabolic activity in the brain regions that were previously dysfunctional. And this coincided with her achieving near-normal cognitive function.
Exactly. The source stresses that this convergence, the dramatic clinical recovery, coupled with the normalization of an objective neuroimaging biomarker, is exceptionally rare and scientifically powerful. It strongly suggests that the PELUT wasn't merely masking her cognitive deficits somehow. It may have actually helped to reverse the underlying metabolic brain dysfunction that is a known hallmark of early neurodegeneration. This kind of alignment between subjective clinical improvement and objective biomarker normalization is a highly compelling indicator of a potential true disease-modifying effect, even if it's just from one carefully documented case. It certainly creates an urgent imperative for larger-scale studies in MCI and early AD populations to see if this remarkable effect can be replicated.
Absolutely. Okay, and finally, let's address Alzheimer's disease, AD itself, the most common form of dementia. What's the state of the evidence for PEA here?
For Alzheimer's disease, the situation described in the review is characterized by a striking contrast. There's a wealth of highly promising preclinical evidence on the one hand, but a critical lack of human clinical trial data on the other.
So lots of lab work, but no patient trials yet.
Essentially, yes. There is an extensive and compelling body of preclinical work studies conducted in vitro, meaning in cell cultures, and in vivo, meaning in animal models of AD, that strongly supports the potential use of PEA for Alzheimer's. Based on this robust preclinical foundation, PEA is considered a prime candidate for clinical translation into AD patients.
What specifically did these preclinical studies demonstrate in terms of effects on AD pathology in the lab or in animals?
They showed quite a range of relevant effects. In cell culture studies, for example, PEA has been shown to protect neurons from the toxic effects of amyloid beta, a peptide. That's one of the key pathological hallmarks of AD. It was shown to prevent ACAN-induced cell death and preserve the function of neurons and also astrocytes, the support cells.
Okay, protective effects in the dish. What about in animal models?
In vivo studies, using animal models like the 3xTg AD mice, these mice are engineered to develop both amyloid plaques and tau tangles. The other major hallmark of AD chronic oral administration of ultramicronized PEA has shown really impressive results.
Such as?
It effectively rescued cognitive deficits observed in these mice on learning and memory tasks. Critically, it also appeared to ameliorate the core neuropathology itself. Studies reported reduced AF formation and also reduced tau hyperphosphorylation, which is what leads to tangles. And aligning perfectly with its known mechanisms we discussed earlier, it powerfully restrained the key drivers of neurodegeneration by reducing neuroinflammation and oxidative stress within the brain tissue of these animals.
So strong, consistent data in models hitting multiple aspects of AD pathology: amyloid, tau, inflammation, oxidative stress, and improving cognition in animals. That sounds very promising.
Absolutely. The preclinical data is described as robust and consistent across multiple independent studies. It provides a very strong scientific rationale for investigating PEA in actual AD patients. However, And this is the crucial point the review makes, despite this extensive and highly promising preclinical evidence. There is a complete absence of human clinical trial results for PEA in patients with Alzheimer's disease cited in the available research.
A complete absence. Not even small pilot studies reported.
That's correct, based on the comprehensive review we're discussing. The literature consistently points to this as the most significant current gap. The conclusion repeatedly drawn is that larger clinical trials are needed, specifically for AD. This preclinical to clinical gap represents the single most important hurdle that needs to be overcome for PEA in the context of Alzheimer's disease, and it's highlighted as the most critical area for future investigation.
Okay, as we've just heard, the evidence across these different conditions is certainly varied. We have high-quality trial data in FTD, compelling but less rigorous findings in stroke, Parkinson's, and MCI, and then this strong preclinical data for AD still waiting in the wings. But some of these outcomes you've described are repeatedly called remarkable in the source material. What does that term really signify in this scientific and clinical context? Is it just enthusiasm or something more specific?
That's a fair question. I think remarkable here isn't just being used as a casual descriptor or, marketing hype. The source uses it, I believe, to highlight outcomes that represent quantitatively measurable, objective effects that suggest PEA might have a capacity to go beyond merely managing symptoms.
Okay.
It implies the potential to perhaps preserve or even restore brain function, or significantly alter the disease trajectory in ways that are currently quite uncommon or maybe even unprecedented with existing therapies for these specific conditions. Let's maybe pull out what specifically makes these outcomes seem remarkable according to the review's perspective.
Yes, let's do that. Starting with the FTD trial results, which seem the most solidly documented.
Right. For FTD, the remarkability lies in both the rigor of the evidence, it being a well-conducted phase 2 RCT, and the sheer magnitude of the effect observed in a disease that currently has no disease-modifying treatment options. Reducing the rate of global disease progression, as measured by that comprehensive CDR-FTD-SOB scale, by over 50% compared to placebo over just 24 weeks.
The 0.53 versus 1.39 change.
Exactly. That difference is genuinely profound. For a devastating disease like FTD, for which there are 0 approved disease-modifying treatments, achieving this level of slowing of decline represents a significant beacon of hope. And as the source suggests, it's a potential paradigm shift in how we might approach treating this condition. So that's remarkable because it directly impacts the fundamental measured course of the disease in a controlled setting.
And then that Parkinson's finding, the one that, as you said, seemed almost too good to be true, improving on the Hone and Yar scale.
Yeah.
Why is that classed as remarkable, even with the observational caveat?
Well, that specific finding from the observational Bertini study, the collective Hone and Yar score improving from a state of decline, 2.5, back to an earlier stage, 1.9, after a year of adjuvant PEA is described as perhaps the single most astonishing result reported in the PEA literature to date, despite its limitation as observational data.
Why astonishing?
Because, as we discussed, standard therapies for Parkinson's managed symptoms, they do not typically reverse underlying disease progression as measured by these objective staging scales. This outcome, if true and replicable, suggests something deeply neuroprotective or potentially even restorative is happening, an effect virtually unprecedented in currently available PD therapeutics. So while it absolutely requires rigorous confirmation in controlled trials, the magnitude and direction of that reported reversal signal is undeniably remarkable in its implication.
And the MCI case study also had that rare alignment of clinical improvement and objective brain imaging data.
Exactly. The power, the remarkability of that single case study on the amnestic MCI patient lies in that convergence of clinical recovery and physiological normalization. The patient's cognitive improvement back to almost normal was significant in itself clinically. But when you combine that with the objective SPECT imaging showing normalized hypometabolism, meaning the brain activity in previously dysfunctional regions returned towards normal, it becomes truly remarkable. The source emphasizes this isn't just masking symptoms. It strongly suggests a potential to actually help reverse the underlying metabolic brain dysfunction that is characteristic of early neurodegeneration. This simultaneous clinical improvement and objective biomarker normalization is rare and provides powerful, suggestive evidence pointing towards a true disease-modifying effect, even at the earliest stages of cognitive decline.
And what about the cognitive gains reported in the post-stroke studies? What makes them stand out?
Well, in the context of stroke recovery, regaining cognitive function is absolutely critical for a patient's independence and overall quality of life. The reported average 2.5-point increase on the MMSE going from 20.2 to 22.7 in just 30 days, as seen in that large open-label study, is highlighted as a clinically significant improvement, particularly given the speed. While it definitely needs confirmation in controlled trials because of the open-label design, achieving such rapid and measurable gains in global cognition, if replicated and proven attributable to the treatment, would position formulations like PEA-LUT as potentially valuable tools for accelerating or enhancing cognitive recovery post-stroke. And that would certainly be a remarkable outcome in neurorehabilitation.
Finally, in ALS, you mentioned affecting survival and function are the most critical outcomes. How are the findings there remarkable?
For a universally fatal disease like ALS, any intervention that affects hard endpoints related to survival and major functional milestones is paramount. The finding reported from that larger study that PEA treatment apparently slowed respiratory function decline and, critically, delayed the need for tracheotomy and death is profoundly significant. These are unambiguous clinical endpoints that directly address the primary cause of mortality in ALS. The ability to potentially extend the time before patients require invasive ventilation and to delay death represents a tangible and deeply meaningful impact on the relentless downhill trajectory of ALS. That makes these reported outcomes inherently remarkable in this specific, devastating disease context.
So, just to synthesize that, remarkable in this context seems to describe objective, measurable effects that suggest PEA might not just be managing symptoms day to day, but potentially altering the underlying disease course, perhaps restoring lost function, or even extending meaningful survival in ways not typically seen or expected with current standard treatments for these specific neurological conditions.
Precisely. It points to outcomes that warrant serious scientific attention and further rigorous investigation because they suggest the molecule, particularly in its optimized forms, might be influencing the fundamental pathological processes or the body's ability to recover and maintain function, potentially offering benefits that go significantly beyond simple symptomatic relief.
Okay. That collection of remarkable outcomes, tempered with the varying quality of evidence, is incredibly promising across multiple challenging conditions. But, as with any potential therapeutic, a critical analysis of the overall evidence picture, the safety profile, and crucially, what needs to happen next is essential. What does the source material say about the overall safety of PEA? That seems like a key consideration. And what are the main future imperatives for research?
Right. A comprehensive assessment absolutely requires tempering the understandable excitement about the potential efficacy signals with a sober acknowledgement of the current limitations of the evidence base, which, as we've thoroughly discussed, vary significantly by condition. However, one of the most compelling aspects consistently highlighted by the review across the board is PEA's exceptional safety and tolerability profile.
How strong is that safety profile compared to, say, other potential treatments being investigated for these conditions?
It's described as one of PEA's most consistent and frankly appealing attributes. Across dozens of studies involving literally thousands of patients, PEA has consistently been reported as very well tolerated, with a remarkably low rate of adverse events reported.
Lower than placebo sometimes.
The review actually mentions a 2017 meta-analysis that looked at PEA primarily for pain conditions. It found that the all-cause dropout rate from the trials was non-significantly lower in the groups receiving PEA compared to the control groups.
Interesting.
It suggests that patients not only tolerate it well, but may actually find it more tolerable than even placebo in some instances, perhaps due to perceived benefits or just lack of side effects.
What side effects are typically reported, if any? Are they serious?
Generally, no. The reported side effects are typically mild and infrequent. Things like minor gastrointestinal upset, a bit of stomach discomfort, or slight drowsiness have been noted in some trials, but they don't appear to be common or severe. And crucially, this high safety profile seems to hold up even with long-term administration over many months, which is absolutely essential if you're thinking about treating chronic neurodegenerative conditions where therapy would likely be needed for years, possibly decades.
And, similar to its efficacy, is its safety likely linked back to its unique mechanism of action, working with the body's systems?
Yes. The source strongly suggests that PEA's robust safety profile is likely a direct consequence of its fundamental biological mechanism as an EYESIGHT. Remember, it acts as an endogenous homeostatic regulator. It gently modulates and enhances the body's own natural protective and anti-inflammatory systems.
Right, not forcing something.
Exactly. Unlike many conventional drugs that might powerfully block or activate a single pathway, which can sometimes lead to unwanted off-target effects and systemic imbalances, PEA seems to work more subtly by supporting the body's intrinsic resolution pathways. This less disruptive, more supportive mode of action makes it inherently less likely, theoretically, to cause significant adverse effects. And that intrinsic safety makes it particularly suitable for the kind of long-term chronic use that would be required in managing neurodegenerative diseases.
Okay, so we have these promising signals of efficacy, including some potentially remarkable findings, combined with what sounds like an exceptionally clean safety profile. But we absolutely must acknowledge the limitations in the current evidence landscape. We can't get ahead of the data.
Absolutely. While the collective data is highly suggestive of broad neuroprotective potential, especially when using those optimized bioavailable formulations, the excitement has to be tempered by a rigorous look at the evidence quality. The source is explicit about this. The strength of evidence varies significantly.
FTD is the strongest.
Correct. The FTD trial data, being from a well-conducted Phase 2 RCT, meets the highest current standards for demonstrating initial efficacy in that specific condition.
But the evidence for the other conditions isn't quite at that level yet, is it?
Not yet. As we discussed, the large stroke study was open label, which carries a risk of bias. The truly remarkable signals seen in Parkinson's, that potential HY scale reversal, and in MCI, the cognitive normalization linked with SPEC changes, come from an observational study and a single case report, respectively.
Useful, but not definitive.
Exactly. While these findings are invaluable for generating powerful hypotheses and showcasing the potential of PEA, they simply cannot replace the definitive evidence provided by large, well-conducted, randomized controlled trials. Clinicians, regulators, and patients ultimately need that higher level of evidence to establish causality and true treatment effect.
And the biggest gap, the elephant in the room, remains Alzheimer's disease.
Yes, the most significant gap identified in the current evidence base is squarely for Alzheimer's disease. Despite that extensive, consistent, and robust preclinical data demonstrating powerful effects on key AD pathology and cognitive deficits in animal models, the review clearly states there is still a complete absence of human clinical trial data reported for PEA specifically in AD patients. This preclinical to clinical gap is highlighted as the single most important hurdle that needs to be overcome for PEA in the context of Alzheimer's.
So, avoiding overstatement and maintaining a nuanced, condition-specific view of the evidence is absolutely crucial for anyone evaluating this research landscape right now.
Exactly. The picture is definitely not monolithic. It requires a careful, condition-by-condition assessment of the available data, acknowledging both the strengths and the current limitations.
Okay, so given these limitations, alongside the promising signals and the good safety profile, what is the clear path forward for research? What do the experts writing this comprehensive review recommend needs to happen next to solidify PEA's potential role?
Well, the consistent call across the research landscape, echoed strongly in this review, is for more and better designed studies to confirm these initial promising results. The good news is that the existing evidence, despite its current limitations, provides a pretty clear and logical roadmap for the necessary future clinical investigation.
Starting with FTD, where the evidence is already the strongest. What's next there?
Yes. For frontotemporal dementia, the success and, importantly, the statistical significance of that Phase 2 trial provide an unequivocal basis for the immediate logical next step. That is, the design and initiation of larger multi-center Phase 3 registration trials.
The big trials needed for approval.
Exactly. These larger trials are necessary to confirm that the disease-modifying effects seen in Phase 2 hold up in a broader, more diverse patient population, and they would form the basis for seeking potential regulatory approval if successful.
Okay. And for those other conditions, Parkinson's, stroke, MCI, and ALS, where the preliminary findings were remarkable, but the study designs were less rigorous, what's the priority there?
For those conditions, PD, stroke, MCI, and ALS, the absolute priority identified is to move beyond the current preliminary data from observational studies, open-label trials, and case reports. It is imperative to design and conduct large-scale, double-blind, placebo-controlled randomized trials.
The gold standard.
The gold standard. These RCTs are critically needed to rigorously test and hopefully replicate those remarkable but still preliminary findings. Can we replicate the potential improvement in the HY scale in PD? Can we see normalization of SPECT imaging alongside cognitive gains in a larger MCI cohort? Can we definitively show a delay of hard clinical endpoints like tracheotomy and death in ALS in a controlled setting? This is what's needed to move these exciting signals from intriguing observations to established clinical effects.
And finally, how do we bridge that major gap for Alzheimer's disease?
For Alzheimer's, the source argues strongly that the time is definitely ripe, perhaps overdue, to bridge that significant preclinical-clinical gap. The wealth of strong preclinical data provides more than sufficient rationale to initiate the first human pilot studies, likely phase 1 safety and tolerability studies, or perhaps early phase 2 proof-of-concept trials, specifically in patients with mild cognitive impairment or those with early-stage Alzheimer's disease.
What would be the focus of those initial AD trials?
The primary focus would likely be confirming safety and tolerability in this specific patient population, establishing the optimal dosing range, and crucially, incorporating biomarker analysis right from the start.
Biomarkers to see if the mechanism we saw in animals actually translates to humans with AD.
Precisely. This would involve collecting data on relevant biological markers, perhaps looking at neuroinflammatory markers in cerebrospinal fluid, or utilizing advanced imaging techniques like amyloid PET and tau PET scans. The goal would be to see if PEA administration influences the accumulation or clearance of the core pathological proteins of AD, or affects markers of neuroinflammation in humans, just as it appears to do in the animal models. This is vital to confirm that the biological effect is actually translating to the human disease before launching into large, expensive efficacy trials.
And across all of these future trials, for all these conditions, the critical importance of using the right formulation, the bioavailable ones, remains paramount, doesn't it?
Absolutely. The source underscores that it is essential, critically essential, that all future studies across all these conditions utilize the optimized bioavailable formulations. Specifically, that means ultramicronized PEA, or the synergistic co-ultramicronized PEA-luteolin, PEA-LUT.
Because those are the ones that worked.
Yes. These are the formulations that have demonstrated the necessary absorbability and achieved the tissue concentrations required to produce the significant positive results observed to date. Trying to run trials using less bioavailable naive forms of PEA would likely undermine the potential to see an effect and would not be a fair test of the molecule's potential based on current understanding.
Okay. So what does all of this mean for you, our listener, who values getting into the real detail and understanding the landscape of potential therapies? We've traced PEA's incredible journey from what was essentially an accidental discovery in egg yolks decades ago to becoming understood as a molecule with a deeply fascinating multi-target mechanism. A mechanism that works by enhancing the body's own systems to resolve inflammation and protect cells, primarily by modulating glial activity in the brain. We've seen how absolutely critical pharmaceutical formulation, things like ultramicronization, is for its effectiveness, transforming it from a poorly absorbed compound into a potentially clinically viable agent. And we've examined the current clinical evidence landscape, which, as we've seen, is complex. It spans from robust trials showing real significant slowing of disease progression in FTD,
Which is huge.
to truly astonishing preliminary findings hinting at potential reversal of severity in Parkinson's and normalization of brain activity in MCI. Alongside that, powerful preclinical data for Alzheimer's that's still, frustratingly perhaps, waiting for human trials to begin.
And all this, as we've really emphasized, comes partnered with a remarkably differentiated safety profile. That exceptional tolerability really sets it apart from many other potential agents being considered for these severe chronic conditions. The history of PEA teaches us a valuable lesson, that the critical turning point for its modern resurgence was finally providing that mechanistic understanding, figuring out the how. The current evidence, while clearly needing significant further rigorous confirmation, particularly through those well-designed controlled trials in many areas, it does provide a clear evidence-based path forward for research, especially perhaps for Alzheimer's disease, where that preclinical rationale is so compelling, but the human data is conspicuously absent.
And here's where it gets really interesting, perhaps stepping back for a final thought. Given PEA's unique mechanism as an endogenous LEME, remember, a molecule that doesn't simply block a single pathway or receptor, like so many traditional drugs, but rather seems to enhance the body's own intrinsic protective, anti-inflammatory, and homeostatic systems. Combined with its exceptionally clean safety profile compared to many conventional pharmaceuticals, could this whole approach represent something more fundamental? Could it signal a shift in how we even think about treating complex chronic diseases like neurodegeneration? Are we potentially moving away from solely intervening on single targets and towards a new paradigm? One focused on supporting and perhaps restoring the body's intrinsic capacity for resolution, for maintaining homeostasis, for repair. That's a profound question to consider as this research continues to unfold. Thank you for joining us for this deep dive.